thank you very much for having a chance to talk our research activity since 2006 by using supercomputer including a K computer and the computer supercomputer at the Human Genome Center, and now Fugaku. Well, I have been involved with the cancer genomics, and the purpose is how cancer occur and evolve and spread and so on. That is we can observe those phenomena, but behind that there are some mechanism, not depending on people. So we want to unravel such very, very complex situation. Okay. And since 2006, we have already developed the series of program on supercomputer for mining a gene network. Gene network, yes, maybe most of the people are not familiar with the biology. Gene network is something like a set of objects. So imagine a company. Yeah, there are many people working. Each people is a gene, and each people is each people's activity is in a sense evaluated as some kind of score. And in the company, there are several kinds of people. Main people are some kind of director. That means some people activate to work some people, and some people disturbs other people's activity. Yeah, it's a complex of pushing more work and disturbing that work. It's a company. So that kind of thing is going in our body. And then what we did was figure out the activity network from the activity score from all people. So this is a typical picture of that. Yeah, some people work, some people disturb, and so on. And the mathematical model we employed and developed is a Bayesian network, not discrete random variables, state space model, structural equation, etc. And all are implemented in Fugaku now. Well, what we want to find is a causal regulatory network as shown here. Some drug is put into some patient, then some gene is affected by, targeted gene is affected by that drug, and then that gene affects the next, that gene next, next, and then phenomena appears. But from single gene, it is impossible to figure out such network. simply clustering, and those are possible. Deep learning is also only possible for such precise classification. Classification shows good observation, but classification doesn't allow us to get into the discovery of mechanism. That is a problem. Well, I want to show more than 10-year our research by using supercomputer about cancer. The cancer, one of the phenomena of cancer is epithelial mesenchymal transition. We call EMT. EMT is, I wrote something, but this is related to tumor grade metastasis, drug resistance, and so on. So it's a very important phenomena. If we could stop EMT in cancer tissues, then cancer is simple. Simply remove that part if possible. Well, I will explain EMT more easily. Well, at the initial point of cancer, each cancer cells are stick to each other, like early stage children. Yeah, parents are guarding those children. So children stick each other. But say at the age of 16, 17, then, well, such children will move at midnight and walk around outside and meet, for example, in the case of girl, meet some boy and talk each other. That means the cancer cells will have an ability to mobility. Then those moving cells will move to other organ via blood vessel, and then living together. And new cancer occurs. It's a metastasis. So this mechanism was known. The key genes are known to some extent. However, its mechanism is still not well understood. This means metastasis is not controlled by drug. This is a strict fact in cancer therapy. And by using the data, more than 10-year-old data that was produced by UK Shanghai Institute, we selected 50 genes which are correlated with the EMT signature genes, and define EMT score. So each cell, cancer cell, has a score with respect to the mobility. It's an EMT score. So these are a list of 50 genes for using our EMT score. It's complicated, and maybe most people are not interested in these lists, but those are important genes arranged here. But even after such 50 genes and 122 EMT signature genes are known, EMT mechanisms are not well understood. Therefore, what we did was the expression of all genes. All genes, number of genes is more than 200,000. And the number of samples is, cancer samples is 762 cancer cells. But these, we evaluated some quality of the measurement. We had to restrict the number of genes from 20,000 to about 13,000, around 13,000 genes, including microRNA. MicroRNA is a kind of, I should say, gene which are not translated into protein. It's an RNA itself will function and very much related to cancer. Expression level is very small, but it affects many genes. And then, when this happened, 2010, around that, the K computer was not available. At that time, we used Human Genome Center's supercomputer. It's a very small computer. At that time, 23 teraflops at peak. And we run, the analysis requires 3 months to reveal each network of 762 genes. Each network here has more than 13,000 genes. It's a very, very complex, very, very complex. And this is a network of low EMT score, meaning do not move. Ability is very low. However, this network has a high ability, moving around. So, it's a chaos. Network of 13,581 is not drawable on display. It's so many. And 762 such big network is, it's something like a very messy ball. So at that time, more than 10 years ago, we have no choice. And therefore, we look at only some specific gene which have been investigated very, very much. It's a E-cadherin. It's a paste which stick the cells to each other. So we thought if E-cadherin expression decreases, then EMT will happen. But that mechanism is not well understood. Very, very narrow view. And then we could not analyze the whole network because of the complexity. So we only focused on the parent genes and the grandparent genes of E-cadherin. It's a very small one. Then, even with that analysis shows how network changes from epithelial cell to mesenchymal cells, simply focusing on E-cadherin. We are very happy to see this phenomena was discovered by our data analysis, network analysis by using supercomputer. Well, but this kind of research is not repeatable because the computing resource is very limited, not limited, but well, in Japan, the available computing resource was none. That is the reason. So we start to analysis at this point. But interestingly, we analyzed the parent genes, parent and grandparent genes of E. cadherin, and we made some scores. Then 24 genes are shown here. And very surprisingly, half of those 24 genes, 12 genes, have been already investigated from the viewpoint of EMT. And the EMT is a very complex system. It's upstream is, well, very vague, and downstream is also vague. But white, not marked genes are not investigated at all from the viewpoint of EMT. Then we asked Professor Takashi at that time, Nagoya University School of Medicine, and he found KLF5. Induces EMT by knocking down, stopping the function of KL5. The EMT process starts beautifully. So new EMT gene was added, and also the mechanism around KL5 was investigated by Professor Takashi. So the EMT is the main topic of cancer research, but supercomputer and mathematics and large-scale data analysis allows us to such discovery. After that, say I added years of discovery. Those genes are related to EMT inducers. So data analysis shows such discoveries after that. Maybe, I'm not sure, all those discoveries are done by wet lab, and maybe those people looked into this list and investigated and validated it induces EMT or not. But I should skip. But this is we've simply focused on E. cadherin, grandparents and grandparents, very short, narrow view. We want to investigate a whole network ranging over 762 cancer cells. And also we faced with the distribution of cancer cells according to EMT score, so not uniform. If the cells are arranged into uniform, it's okay. However, for example, drug resistance data, it's not uniformly arranged. So the Professor Park, she is now the professor at Data Science Center of the Tokyo Medical and Dental University. And she collaborated with the Fujitsu laboratories, artificial intelligence laboratory. Dr. Maruhashi, he devised the network analysis tool by using deep learning by so it's a deep tensor. An input of deep tensor is the network structure. Tensor analysis is also included in that. And then I thought, can we combine the network analysis and deep tensor analysis so that we can reveal the EMT mechanism more. Furthermore, such phenomena appears not only cancer, but also all area of biology we face with such phenomena. And we have been working more than 3 years on this, and we developed the explainable, interpretable AI, and last year, started the Fugaku project. After Fugaku project started, we implemented deep tensor and also interpretable AI, we call TRIP, on Fugaku. This is, Japanese. The deep tensor plus interpretable AI TRIP revealed from such 762 very complex network, and found it is published in PLOS ONE last November, and what I want to say is from the 10-year-old data of cancer, the deep tensor plus TRIP unraveled the biological fact related to EMT, which were discovered in the last 10 years instantly. We are very much surprised. All kinds of, not all, and some of the discoveries found by deep tensor implemented on Fugaku shows uninvestigated pathways and genes. Those are next investigations. Well, my message is 10-year activity discoveries produced by 10-year activities by molecular biology, cancer biology are unraveled from 10-year-old data by using deep tensor, TRIP, and also network analysis should be done. Network analysis by using Fugaku. Well, artificial intelligence is the next big player in genomics. And we are now, this is a preliminary study. We are now going to clonal network analysis, how cancer happens and develop. So it's a kind of network, and the mechanism is completely unknown. So by using this explainable AI on big data analysis, we want to get into this pioneering research area. Thank you. So this is a clonal evolution of that. Yeah. That's it.